Hello, and welcome to the Logos Research Associates live stream broadcast. I'm Dr. Andrew Fabich, and it's my privilege to be your host for this presentation. Logos Research Associates is a fellowship of scholars and scientists that are committed to upholding a high view of science and a high view of Scripture. We sincerely believe that scholarship, logic, and the scientific method can and should be used to show that the Bible's historical claims are both credible and ultimately superior to the secular model of origins. Logos sponsors many creation research initiatives and projects involving many different experts and scientific disciplines, all working towards the goal of increasing the explanatory power of the creation model of origins and of Earth history. The goal of this live stream series is to provide you with the opportunity to hear from top researchers who will convey cutting-edge scientific evidence for the creation account. Before I introduce our speaker to begin our presentation, we will open in a short prayer if you would please join with me. Heavenly Father, thank you for this evening that we can get together, Logos Research Associates, for getting together this group of scholars, and in particular, Dr. Wilson tonight. Thank you for the friendship that we have together and the fellowship that we can have taking a look at your creation. I ask that you would use this evening to use Dr. Wilson to speak through this screen and reach the viewers on the other side, that they would be encouraged, and it would be an encouragement to him as well, and that we all would just take a stand back in awe of all the work that you have done here for us to enjoy. And so we ask you all of this in the great name of Jesus. Amen. I now have the pleasure of introducing our speaker. Dr. Gordon Wilson is currently a senior fellow of natural history at New St. Andrews College in Moscow, Idaho. Prior to New St. Andrews, he was on the biology faculty at Liberty University. Gordon received his PhD in environmental science and public policy from George Mason University in 2003. His dissertation research focused on the reproductive ecology of the eastern box turtle. He also earned his master's in entomology in 1989 and bachelor's in education biology in 1984 at the University of Idaho. He has written over 30 popular articles for Answers Magazine, Answers in Depth, and their online content. In 2015, he published a biology textbook called The Riot and the Dance. He's also the narrator of a nature documentary series called by the same name on Angel Studios and Canon Plus. In 2019, he published his book, A Different Shade of Green, A Biblical Approach to Environmentalism and the Dominion Mandate. In 2023, he published Darwin's Sandcastle, Evolution's Failure in the Light of Scripture and the Scientific Evidence. And now we welcome to the broadcast my good friend, Dr. Gordon Wilson. Welcome.
Thank you, Dr. Fabich. It is so good to be here, and I love giving this talk. Just a couple words about the title, Magnificence of the Mundane. First, I just like alliterations, but secondly, I like the paradox that we see in the title. Now, obviously, it's sort of oxymoronic if we take both literally. An oxymoron, as one definition puts it, is a figure of speech which apparently contradictory terms appear in conjunction. And so I should put quotation marks around mundane, because if you look at those two terms, magnificent means impressively beautiful, elaborate, or extravagant, striking. And mundane is the exact opposite, lacking interest or excitement, dull. So I like this paradox. The Apostle Paul in 2 Corinthians 6, 10, it's a familiar passage. He uses a similar way of bringing two seemingly contradictory terms together: sorrowful, yet always rejoicing; poor, yet making many rich; having nothing, yet possessing everything. So it's kind of getting at the same thing. It causes somebody to, their curiosity is perked. What do you mean, magnificence of the mundane? How can that work? Well, first of all, the mundane is not what, it's more of an issue of the observer, not the thing observed. Because what I'm going to discuss tonight is a handful of things that are common We might call them mundane, but that is an issue with our observational skills. Our curiosity has grown calloused, and our we've got cataracts over our wide-eyed wonder that we used to have as a kid. Our eyes are sort of covered over with a cataract of indifference that we can't see the beauty and the glory of God's creation. So what I wanted to do is just kind of cover a handful of a few animals and a few plants. I could do a lot. I could go on and on and on, but more is not better. I wanted to focus on just a few things and showcase the glory and the magnificence of what we often overlook because they're simply common. And that, again, the mundane is a problem with the observer, not with the thing being observed. So what I wanted to do is look at just a few animals and plants. As far as the animals go, I'd like to look at box turtles. Dr. Fabich had mentioned that I did my PhD on the eastern box turtle, but it was on a totally different topic. I'm going to be looking at the freeze tolerance of box turtles. And then I'm also going to look at water striders, jump into entomology. I'll look at their walking on water and their hunting skills. Both of these are common. Well, at least the first one, if you're in the eastern United States, you'll see box turtles and some places in the West. But unfortunately, out here in Idaho, we've got one species of turtle. It's a bummer for me, but that's okay. There's lots of other things that are magnificent. I'm going to look at orb weavers, which is a family of spiders, and look at their incredible web design. And then houseflies, escaping the puparium. You might wonder what a puparium is. We will discuss that when we get there. As far as the plants go, I'm going to look at mosses. I taught botany for 10 years and at Liberty University, and I teach it as an elective here, and I just love plants because they have all sorts of surprises, and their spore dispersal is incredible. I'm also going to look at ferns and their spore catapults, and exactly what I mean by it's exactly that, a catapult. Now, some of you who are biologists are familiar with some of these things, but you probably haven't seen them presented in quite the same way. And hopefully I will set the hook and get you out there looking. And then lastly, as far as plants go, I'm going to look at dandelions and their fruit parachutes. And more on that later. But first, let's look at box turtles. Now, just wanted to start with a story. When I was a kid, I grew up in Annapolis, Maryland. Across the road from us was a woodland, and I was just fascinated with box turtles. I think that's what set the hook for me in biology, is just seeing box turtles when I was a real little kid. When I came out west, there was no box turtles, at least in Idaho. And I asked a friend when I was in high school or late junior high, would you go back and get me a box turtle and bring it out west? And he did. He came back, and it was a beautiful box turtle. I named him Waldo Pepper. I kept him in my backyard and had a pen for him. And then I thought, well, they can handle the winter, snow and cold. So I decided to, during the fall, I piled a bunch of leaves on top of Waldo, and he dug down a little bit, and I put a tarp over it, but I didn't warm it at all. I just let, other than the leaves, he was pretty exposed to the elements. And that winter, it was in the mid to late 70s. It was brutal. It got down to 30 below that winter. And I was just thinking, even in my teenage years, I was thinking, oh man, that my Waldo Pepper is toast. And in the spring, when it was the first warmish day, I scraped the leaves out and I saw the top of his shell. He didn't dig down very deep. The top of his shell was like at ground level, and I touched it, and he moved his head and jerked, and I saw his shell roll a little bit, and I realized he was alive, and I was just shocked. I was flabbergasted that he could do the 30 below for that cold snap. And so, how does that happen? How do they do this? Well, first, there's several reptiles that can do it. There's garter snakes. There's probably more, but garter snakes, box turtles. As far as amphibians, wood frogs, I'm sure that list will expand as they do more and more studies of high latitude herps. What they do as the days get shorter and the days get colder, the liver starts to crank out glucose, and they're probably breaking down the glycogen in their liver and then releasing it into the bloodstream. And the glucose is then taken up by the body cells and incorporated into the cytoplasm of the cells. And they also pump water out of the cells, move water out, glucose in, and basically just cryoprotect the cells and prevent ice crystals from forming inside the cells. But outside the cells, between the cells, in the body cavities, around the organs, that can freeze solid. So basically, you've got ice throughout the body, around the brain, around the muscles, around the lungs. And as long as the intracellular environment stays liquid, it sort of stays, it's alive, but for all intents and purposes, metabolism is not even a pilot light. What's a real kicker is that the heart stops. Their heart, I'll say it again, their heart stops. And it can stop for a long time. And I don't know what kind of metabolism is going on, but it's pretty minimal, especially when their heart is flatline. And when the spring rolls around and the ice starts to thaw, and the ice leaves the body, the heart starts back up again, and they're alive. So it's a great metaphor for death and resurrection. It's incredible. So even though I didn't do my work on that, that's box turtle physiology. I did it on reproductive ecology, but still, box turtles are amazing. Okay, let's switch gears to entomology. I can't think of something. Well, there's other more common things, but everybody's familiar with water striders, these insects that walk on water. And I wanted to just kind of unveil what's going on there. These insects are in the order Hemiptera. They are a true bug. They've got piercing, sucking mouth parts, but their body is covered in what is called a hydrofuge. They're called hydrofuge hair piles. And basically, that means that coming out of their exoskeleton, out of their cuticle, are hydrophobic microhairs. And there can be several thousand per square millimeter. And it basically just gives them this waterproof coating, very, very thin, low pile. And so if you splash them, the water just, the saying, water off a duck's back, you could say water off a water strider's back. Water just beads off of them. They stay dry because of their hydrophobic hair density. Also, they're lightweight. And notice how their mid and hind legs are sprawled out so that they're distributing the weight over more surface area rather than on their tiptoes. It's more like a cross-country ski. It's laid out on the water. And so the property of the cohesivity of water. Basically, we call it surface tension. The water molecules are hydrogen bonding, staying close together, wanting to huddle, and it takes a bit of force to break through that surface tension. Plus, they distribute their weight across a broader surface, as well as the hydrophobic hairs, and that allows them to just scuttle across the water with impunity. Now, they use their middle legs to scoot across their water, and when they push their middle legs down, it forms a hummock of water behind, and then they can kind of push off of that and move forward. Also, their back legs are used to steer them. Their front legs are pretty short, and they're kind of like miniaturized praying mantises. They're raptorial forelegs that are used for grasping prey. They are predators, and so this is wanted to just talk a little bit about their hunting skills. They when an insect, hapless insect, lands on the water surface, it's struggling, and as it's struggling, it sends ripples across the water, and the water strider senses those ripples, and their legs go up and down when the ripples hit them. Now, if they're oriented so that their head is facing kind of towards the prey, their front legs will be resting on the water. And if they're facing, they can't see the prey because it may be too far, but they sense the ripples. And if the ripples come at them straight on, their legs will go up and down together. If they're at oblique to the prey and the ripples are coming this way, let's say then their right leg goes up first and their left leg goes up second, so they can adjust. And as long as their legs are kind of bouncing up and down together, they're on the right trajectory. And so they scoot forward until they run into the prey. So by changing their orientation based on the ripples, they can move in the right direction. They get close enough, then they can see with their compound eyes, and then they pounce. And when they pounce, they grab the prey with their forelegs. And like I said, they have piercing, sucking mouth parts like all true bugs. Their piercing, sucking mouth parts are kind of like, if you look up here in the upper right-hand corner, they've got this long beak, which is segmented, and that's called the labium. And I'm going to use an analogy here. A labium is kind of like a long segmented hot dog bun, and there's a groove, an open groove on the front edge, and the stylets are like very, very skinny hot dog running in the groove, like a hot dog in the bun. So the labium is the bun. And if you see this cutaway here, you can see the stylets. Those are the things that actually pierce into the prey. The beak, as those pierce in, they come out the tip, the beak bends backwards as the stylets get poked through into the prey. Now, if we do the cross section of this beak, you'll see the maxillae, which are paired mouth parts that form two grooves. And when they're pressed together, those two grooves form channels. And then to the outside of those, we've got the mandibles, which help cut into the prey as well. The tips of the mandibles and the maxillae do some cutting. But once they poke into the prey, they can inject saliva down into the prey for pre-oral digestion. And once it's been digested, then they suck it up the food canal. So there's just a lot of cool anatomy. There's a lot of cool behavior. And then the hydrophobic hairs to allow them to walk on water just makes water striders magnificent, even though they seem mundane. Let's move on to orb weavers. Orb weavers are very familiar, again, very common. So common, we might even think they're mundane, unless you're a little kid or an arachnologist. I remember as I was going through my box turtle site, it's pretty cool to see an orb web if it's a nice flower garden and it's lit up with the sun. But if you're trudging through your research plot and the web is, for all intents and purposes, invisible because the light's not reflecting on the silk, and then you just face plant and your face goes right in the middle of the web and the web just wraps around your head, and the response evoked is mostly ew, ick, a spider web. But if we stand back and we look at the intricacy of this, these are marvelous structures. There's about 3,000 species of orb weaver spiders in that particular family. And obviously there's going to be differences in size and shape, and some details and nuances are going to vary between species. But the general pattern is pretty incredible. Let's just look a little bit at the anatomy of the spider with regard to their spinning. Now, if you look here at this gray silhouette of the spider, you got the abdomen, you got the cephalothorax, and the pedipalps and the chelicerae for the fangs and all of that good stuff. But at the end of their abdomen, we've got what are called spinnerets. Now, spinnerets are basically tiny conical gadgets. I would say that's similar to your garden nozzle, spray nozzle. Think of the spray nozzle as a spinneret. And then you've got lots of little holes in that spray nozzle, and we could think of those as the spigots. So sometimes over 1,000 spigots on a spinneret. And the spinnerets, there's more than one. They're attached to up to 7 different types of silk glands. You've got here, they're all highlighted in different colors. You've got the egg case silk glands. You've got the sticky spiral. We'll look at the temporary spiral, framework, wrapping silk, all these different types of silk. Now, basically, these spinnerets can dispense the liquid silk in glue gun fashion. Now, we used to think that when it came out, it turned solid. It was a liquid silk. It turned solid because of exposure to air. But we found out now that the silk protein, it's not reaction with air that causes it to solidify. As the legs, kind of like when you take a glue gun and you pull, you see a string come out, or like if you're building a model airplane and you're bad at it, and you put a daub of glue and you pull away too quickly, you see a big glue string. Well, the spider actually pulls the liquid silk out, and as it pulls it out and stretches into a thread, the protein fibers start to line up and get more and more parallel. And when they line up, they hydrogen bond with each other, and that turns the liquid into a solid, albeit a stretchy, flexible solid. And so I'm just barely, this is just bare bones. It's more complicated than this. Now, the spigot designs and the glands, the different spigot designs and the different glands make threads that vary in thickness, stretchiness, and stickiness. Okay, now in most cases, as I said, the legs pull out precisely the right kind of thread from the appropriate spinneret at the right time to weave a geometric masterpiece. The whole job generally takes less than an hour, give or take, and is finished with mind-boggling dexterity and speed. And often they tear it down and build a new one for the next day. They don't always keep it. They don't just build a new one because somebody trashed it. They often take down a web and build a new one the next day. So in a complicated series of movements, it constructs the frame and mooring threads. Let's look at a picture that diagrams this out in steps. Now, here's just a simple pattern. You've got the frame, mooring threads that hook it to whatever it's building its web on. So you've got your frame, your mooring threads, and then you've got radial threads. And they put in a few radial threads to get this orb web started. Then they add more radial threads, and because the frame and the mooring threads and the radial threads being the spoke-like threads, they have to be able to support the entire web, as well as the weight of the spider and the weight of the prey. And these threads need to be stronger, thicker, and non-sticky. So that is stronger, thicker, and non-sticky. And then while they spin more radial threads and fill out all these spokes here, the orb spider cross-laces the radial threads near the center of the web, and this is called the hub. Okay, so that if you have the patience and you're going out at night with a flashlight and you're willing to stay up for a good bit, you can watch them build it. The next stage is that they build an make sure I can pronounce this auxiliary let's just say a temporary spiral, an auxillaria spiral. Now that's also strong and non-sticky, and they start from the center and move out. Once they've done this auxiliary web or auxiliary spiral, then the last thing they do is the sticky spiral. And the auxiliary web, they started from the center and moved out. In the sticky spiral, they start on the outskirts and move in. And these guys are much more closely spaced. And they use the auxiliary web as a guideline. And as they're putting in the sticky spiral, they take down the axillary spiral. Now, what's amazing is that their dexterity of all 8 legs, they're stepping on all of the non-sticky threads, and they're keeping their legs out of the sticky ones. They don't want to be trapped in their own web. And talk about fine motor skills. So they lay down this sticky spiral using that auxiliary web as a guide. And then the sticky spiral is exceedingly thin. The web is exceedingly thin and it's stretchy, and they put a little bit of glue on it to make it sticky. And basically, after they're done with that, they take their position on the web with their legs positioned so they can sense a vibration if prey hits. They also leave a little bit of a gap between the hub. Once they're done with the sticky spiral, they keep a little gap so that the spider can slip through from one side to the other really quickly if the prey gets stuck on the side opposite them. So they can switch sides without having to go all the way to the periphery. So talk about amazing. I need to sit out one of these. It's only an hour long, and I need to sit out and watch one being built. It would be a great exercise. One of my favorite things about insects, you think, well, you can't get more common than houseflies. This is definitely common. And I wanted to talk about how houseflies escape from their puparium. And you might want to know what a puparium is. Well, if you know a little bit about the life cycle, these insects exhibit complete metamorphosis, and they hatch first from eggs. The maggot comes out of the egg, and the larvae are called maggots. They go through several instars, molting into a bigger maggot, bigger maggot, bigger maggot, till finally they get to be the full-sized maggot, and then they have to go through metamorphosis. Well, they don't spin a cocoon. So what they do is they molt and they form a pupa. But the pupa forms inside their last maggot skin. So even though they have this pointed, they point these and call them pupae, this brown case is really a puparium, and the pupa is inside. Now, the puparium is basically the last maggot skin. It kind of becomes an oval. It gets dry and crispy and forms a little mummy bag for the fly pupa to do its thing and do its metamorphosis. Now, when the fly becomes ready to emerge as an adult, it has to it's somewhere hidden. We don't see this happen. They've got it on film. You can probably find it on YouTube. They come out of their pupal skin, and that's a pretty easy thing to do, because the pupal skin is pretty thin, but the puparium is tough. And right near its head, it's got a thin seam, sort of pre-weakened seam right around the head. And the fly has to push against that cap and pop out of the puparium. So it's encased in their mummy bag. Now, just pushing on it isn't enough. This whole group of flies, including houseflies, blowflies, which include blue bottles, green bottles, tsetse flies, and a number of other flesh flies, all of them belong to this one group called Schizophrida that produces a thing called a telinum. Now, the telinum is just amazing. Not all flies have it. Only this group of flies. Now, when they're pushing against the puparium's cap and they can't exert enough force to pop the cap, muscles in their body contract and move the hemolymph, or the blood, up into their head. Talk about a head rush. There's a seam right here between their compound eyes, kind of a parabola seam right here. And when the blood flows into their head, the trap door on their face just kind of pops open, and this thing that looks kind of like a driver's side airbag comes billowing out of their face. But it's not an airbag, it's a blood bag. It's a hemolymph bag. It billows out of their face and exerts that balloon, basically called a telinum with a P, a silent P on the front, exerts pressure on the inside of that cap and pops that cap off. So here's the puparium, and here is an adult fly. It's still got very light off-white skin. The telinum has billowed out of its front of its face, exerted pressure on the cap right here, pops it open, and now wiggles with undulations, wiggles out of this very, very tight mummy bag. After he gets out, the blood pressure in the telinum goes back to normal. The telinum deflates, goes back inside the face. The plate that was covering it all up, clicks back into place, and then it hardens its exoskeleton and then flies away to land on some refuse and then go land on your cheeseburger. So I'm not trying to make you feel bad about swatting flies at all. Go right ahead. I just want you to know what amazing engineering has been put into that fly before you kill it. Let's change gears into plants. I am very fond of plants because, well, most people like animals because they do things, but plants do too. You just have to look a little closer. Okay, first, mosses. And I'm going to look mostly at moss spore dispersal. Just a little bit of background on mosses. You'll see these leafy green carpet, and that's called the gametophyte stage, which shows alternation of generations. So you got the leafy gametophytes, which mean gamete plants. And then you've got and they are haploid. They will be either male or female, and the males have usually at the top of the moss carpet fiber, they have little sperm packets called antheridia. And so mosses don't have flowers, they don't have seeds or anything like that. But the gametophytes make gametes. You've got the female moss that makes archegonia, usually at the top, but sometimes other places. And inside the archegonia is a little tiny egg, one egg. The sperm packets, the antheridia, are packed with lots of sperm. When it's the right time of year, the rain saturates the moss, the antheridia break open, the sperm are flagellated, the sperm swim through the water film and swim over to the female moss and swim down the neck of the archegonia and fertilize the egg. And now, to use familiar terms, the female moss is pregnant. It's got a zygote there, and that zygote is now diploid, whereas the gametophyte is haploid. The zygote is now diploid and starts to grow out of the archegonia and forms this long, slender stalk. And this is a completely different generation than the moss gametophyte. This is haploid mom, and this is diploid junior. So there's a stalk, and there's this knob at the top called a sporangium. And so we call this a sporophyte because it's a spore plant. We call these gametophytes because they're gamete plants. Now, inside this sporangium, which means spore container, it needs to make haploid spores. Well, in order to make haploid spores, you need to go through meiosis. So inside this sporangium, it goes through meiosis to make a bunch of haploid spores. Here's a zoom up. And what's amazing about this is very subtle. And we just look at this stuff all the time. We see moss all the time, shaded sides of buildings or up on rooftops. But we don't look. It just kind of blends into the shrubbery, and we don't look at the intricacies and the details and the complexities. So what's going on here? Here's this sporangium zoomed in. Let's use an example of a pepper shaker, a salt shaker. When you want salt to come out, you flip it upside down and salt comes out. Now, it comes out at first, but then it stops and then you shake it. Well, moss can't shake, but they've got other tricks up their sleeve. So first, meiosis occurs inside this, makes a bunch of haploid spores, up to 50 million or so. And there's a pre-weakened seam that forms around this moss capsule or sporangium. And when the spores are ready to come out, this cap loosens and just pops off. Almost like it was planned, I say facetiously. Okay, it pops off, and then just like a salt shaker, the spores come out. Notice that this is also, when it first grows up, it's kind of straight up. But then when it's about ready to dispense the spores, there's a nice little hook that forms in the stalk to allow the spores to fall out by gravity. When the cap pops off, there's another cool trick. Yes, they're nodding so that gravity helps spores fall out. Inside, there's up to 50 million. But there's also, in some mosses, not all, one group of mosses has a thing called a ring of peristomal teeth. Peristomal means around the mouth. And these are very, very thin ribbons that are tapered, have tapered tips. And when it is moist, they curl inside the sporangium, because the sporangium doesn't have the luxury of shaking like a salt shaker. So these guys curl inside, and then tips of these peristomal teeth get dusted with spores. And then when it's dry, they unfurl and help throw the spores out. Pretty amazing. And this is happening all around us. And look how small this is. This is my thumb here. And this is the gametophyte, sporophyte, sporangium. And there you have it. Now moving on to another magnificent plant. Spore catapults. Now, some of you, if you are a biologist, you might like to look at things up close and see on the undersides of fern fronds, often there are spots. Now, the spots can be round, dome-like. They can be linear. They don't have to be round. There's a lot of diversity in ferns. Same with mosses. I forget the count on ferns, but mosses is over 22,000 species. So ferns are up there in the thousands. And so we've got these spots, and these spots are called sori, or sori if you're a Latinist. The sori, or sorus singular, let's look at a sorus. One dot is actually a cluster of sporangia. So that's one sorus, and there's dozens of sporangia all clustered together. If we zoom in on this sorus and go up to one individual sporangium, let's pluck one sporangium out of that little dot. Now that dot might, here in this case, this dot is almost 3 millimeters. That or almost 4 millimeters, almost 4 millimeters. And this, if you pluck one sporangium out, that's what it kind of looks like. You've got a stalk connecting it to the leaf. And let me just use another analogy. Think of the stalk as a tee and a golf ball on the tee, and then a millipede wrapped around the golf ball. That's sort of architecturally the shape of things. Now, this thing that I referred to that looks like a millipede wrapped around a golf ball is called an annulus. The sides of the sporangium are very, very, very thin-walled. And this is just an incredible structure because it is literally a microscopic catapult. These cells, once they form, they become dead, but they're water-filled. And when the spores are ready, and the spores are made by meiosis, meiosis makes them haploid, water starts to evaporate out of the annular cells. And as they evaporate, it causes tension, and the annulus wants to peel backwards, wants to, if we call that a millipede, the millipede wants to do a backbend. And when it does, as the water evaporates, the annulus peels back and it's kind of inside out, or everts is the biology term is. It peels back, the walls rip away, the lip cell rips here, and the thin walls of the sporangium form basically a nice little cup that holds the spores. As the annulus peels back, as water evaporates, it forms air bubbles in these annular cells and causes cavitation to occur. And eventually, I don't know the exact mechanism, but that cavitation causes the annulus to snap back to its original position. Now, it peeled back slowly, but when cavitation happens, it snaps back violently and carries its basket of spores with it and flings the spores out. But this happens at the right time when the spores are ready to be dispersed. Now, I wanted to just show you, here's a glass slide of a fern leaf. Here's the fern leaflet with the upper epidermis, lower epidermis, and a bunch of sporangia. These are the stalks of the annulus. So again, this annulus peels back, and then when it gets to a certain point, wham, it flings the spores violently. Amazing, wonderful little gadgets, beautifully designed. And when people go into the nitty-gritty of this, they're constantly using the word design and ingenious, things like that. So let's look at a quick, let's see here, video. Let's see here. There we go. This is showing the annulus coming back, this out of a microscope, and then wham, throws the spores. Here is another annulus peeling back, cup of, and then threw all those spores out. So you can see the thin wall of the sporangium on either side of the annulus. See what I mean by looking like a millipede wrapped around a golf ball? But when it opens up, the size of the sporangium form a cup. So there you have it. And last but not least, dandelions. I can't think of something more familiar. We are all familiar with dandelions, little weeds that grow in people's yards, and we often try to kill them, but usually kids find them very fascinating. They pluck them up, especially in this stage, and they love to just blow these little seeds. Now, you might be wondering why I call it a fruit. Technically, it is. These are many flowers on a dandelion. This is a head type of inflorescence. And each one of these, which we call a seed, is actually a dry fruit with a ovary wall, and there's a seed inside it. So it's not wrong necessarily to call it a seed because there's a seed inside. There's a nice little stalk, and there's a nice little parachute. Now, to use the proper terms, this little parachute on this dandelion fruit is called a pappus. And a few years ago, a journal article was accepted by the journal Nature, and they were basically describing a totally different type of aerodynamic drag that wasn't familiar in all of their aerodynamic studies. Now, people into the physics of it think of air as a fluid. It's just a very low density fluid, but they put these guys in a wind tunnel or some kind of tunnel so that they could image the vortex that this pappus creates. And this pappus, this little tuft of bristles, is around 100 bristles, give or take, sometimes down to 90, some up to 110, but it's right around 100 seems to give the best kind of drag. Now, what's amazing is that it is like a parachute, but not like a parachute. Your parachutes don't have bristles; they have a whole membrane. It's a enclosed disc. Now, they discovered that this pappus creates a vortex called a separated vortex ring. Most in aerodynamics, airplane wings form a vortex that's attached to the back of the wing, that the wing drags through the air. And same with a bird wing. But this is a separated vortex ring, and that it's detached. It's not attached to the pappus itself. There's a little gap here, and that's why they call it a separated vortex ring. What they discovered is that this pappus, this vortex ring gives the pappus 4 times the drag per unit area of a solid disc. So in other words, if we had a solid disc that was equivalent in terms of area of the chute, it would be a much smaller disc, but it would be covering the same amount of area. Well, this pappus creates 4 times the drag than this per unit area of a solid disc. So it's basically the most bang for the buck. You've got this exquisite design that creates this totally new stable vortex ring that this descending dandelion fruit drags through the air, slowing its descent, and it's economical because they don't have to put as much material into the pappus. So it's lightweight, it minimizes the material use, and as well produces a stable separated vortex ring to increase drag. So maximal drag for the least amount of pappus. Just brilliant. And they're still studying this thing. Such a seemingly simple structure, and yet I just read today that the weather even affects the angle of the pappus bristles. So if it's a dry, windy day, the pappus opens up more to seize the opportunity to catch more wind. So it can go farther if they open up the pappus a little bit more. So it can make it's not choices, but it's literally designed to catch wind when it's there better, because it can change the angle of the pappus bristles. So in conclusion, just a couple take-home thoughts. First, if you find yourself kind of flagging in your curiosity, when I initially described often we've got this problem of the law of diminishing returns. The more common something is, the more bored we get with it. And it's sad, but true. It's like it happens all the time. It's sort of part of how we view the world. If we see it a lot, we just kind of grow bored with it. And an antidote to that is to spend some time observing the creation in detail. Loosen up your schedule. Sometimes we're just so packed, so busy. We might be super curious about our wheelhouse research, and this is what we love. But we need to, if we want to reinvigorate our curiosity and take off those cataracts that blind us to that wide-eyed wonder of childlike curiosity. Observe the creation in detail, because then we start to see the intricacies and the complexities. Ponder that God made it. I mean, yeah, that evolution random chance, time, chance, matter, energy does not create this incredible complexity. No amount of time can do that. And uh, and give thanks, because God is the one who uh, is the engineer, the artist behind all of these little things. And you don't have to go on a big safari in Africa or Australia to see stuff. All you have to do is have a magnifying glass, or even better, a dissecting scope, and go out and explore your backyard. It's there. Now, as my brother likes to say, it's a get to, not a got to. I'm not trying to bind your conscience and make you feel guilty if you don't go out and start crawling around in the weeds. Um, but if you want to, again, renew that childlike wonder, then start observing creation in detail. A couple verses I wanted to end with. It is the glory of God to conceal things, but the glory of kings to search things out. God didn't drop all of these amazing things out of the sky in shrink wrap textbooks or encyclopedias. We had to figure it out. We often just think, oh, somebody else did, but there's so much more to be discovered out there, and all we need is just a lot of citizen scientists that are going out. They don't have to be technically trained. They just need to have a sharp eye and curiosity. A lot of our first pioneer scientists, they were intelligent, they were good observers, and they were desperately curious about how things worked. And that is sort of the impetus behind the scientific enterprise. Be just curious about things and keep that wonder alive. Now, you might say, why the kings? Well, kings back then seemed to be the only people that had the leisure time to search things out. Most people were hand to mouth, and they had to work hard to get food on the table, and kings were the ones that had a little more time to search things out. But now, with all of the amenities that we've accumulated and all the technologies that we've accumulated, now that privilege is not just for kings. It's not just for scientists. It's for anyone with a sharp eye and a curiosity. Psalm 111, verse 2, great are the works of the Lord, studied by all who have pleasure in them. That's a great verse. Notice it's not a guilt trip there. It's studied by all who have pleasure in them. So if you don't have pleasure in them, it's not your thing, and you'd rather just study your thing, great. But if you have pleasure in them, great are the works of the Lord, and study it. Follow your curiosity. And lastly, finally, brothers, the Philippians passage, finally, brothers, whatever is true, whatever is honorable, whatever is just, whatever is pure. And notice a lot of this we think of as just moral, it's only moral things, or it's just the Bible. Well, no, whatever is lovely, whatever is commendable, if there is any excellence, if there is anything worthy of praise, think about these things. NIV says, dwell on these things, ponder these things. Go out and explore. God gave us senses. He's given us a mind. He's given us curiosity. He's given us eyes, touch, hearing. There's so much out there, and don't grow bored with it. I say in The Riot and the Dance, to be bored in this world is to be boring in this world. So, that's it for my talk. I am ready for any questions. I'll take it, let you have it. Um, Dr. Favich.
All right. Wonderful talk, Gordon. I really enjoyed listening to it. Was reminded of so many things, but I don't want to miss out on the guests that we have with us and some of the questions that they had. from Andrea. She had a question that had to deal with the spider that you described, as far as the building of the web and the winged ants that came out later in the day. Do you think the spiders smelled the pheromones? I think it adds another layer of complexity. It's not just the
Hmm.
building of the web
A
there.
lot of spot. Well, yeah. Oh, okay. Building the web at that location.
Yeah,
Boy, that's a good question. I think they definitely build the, you know, are going to get uh, some fly, a fly zone where you've got not just winged ants, but um, all sorts of insects flying through. So um, I'm not sure if one is stimulated the other, but they definitely build their webs in strategic locations.
it's pretty amazing. I was looking.
I don't know. That would require
Yeah,
I don't know if the spiders smell the pheromones. That would be pretty nitty gritty literature search to figure that
yeah,
one out.
yeah. So another question about the spider from Fred. He asks, how much of a spider's daily energy needs are spent in making a web? I usually go around webs unless they're in my screened room.
Boy.
The process of making that silk and
Wow. Uh, well, it's amazing how I don't know the mass. The threads are amazingly thin. The sticky spiral is very thin. The mooring threads, the frame, all of that is thicker, but still um, thinner than you might think. I think the sticky spiral, the diameter of the thread is in the micrometer range. Um, Sometimes down to like 2 micrometers, like the length of E. coli could be the width of a spider web. So it's not a whole lot of mass, but when they take it down, they will recycle the fibroin, which is the silk protein. They'll eat it, and then they can use all of the nutrients and the amino acids to build um, another web for another day. But again, I don't know what the mass is, but I think it's pretty light. But again, I don't know what percentage of the body mass is the silk.
yeah, I think that's pretty amazing. All the intricacies of the spider and its web, and over in Proverbs 30, we oftentimes say to look at the ant, but
Well,
King Lemuel said to also look at that spider's web to learn from it.
right. Oh, yeah. I was actually sorry. You were saying daily energy needs. I would say, yeah, I was thinking mass, but the energy needs. I would say a lot of it goes into doing the web because really a lot of the spider's activity is doing that because once that web is done, um, they just sit there and wait for a catch. And then some energy is spent dexterously winding up their prey, using silk for that. And then, and but yeah, I would say a lot of their energy goes into building that web.
Pretty
Next
amazing.
question.
Yeah. So we have storytelling. Ron asks if this is going to be your next ride in the dance series. Please.
Well, I am not the director, so we I just go along for the ride. I um, the they decide where to, I mean, I can give input, and I have given input in this regard because often right now um, the the tendency is to go for the exotic which is fine. That going to Africa, going to Australia, that is that that always increases. Oh, what's the term where people that increases the ratings when you see elephants and tigers and big charismatic megafauna. But I have put it in the director's ear that I think we could really do an incredible Magnificence of the Mundane type of Riot in the Dance episode, where we zoom in with macro lenses and film stuff that in some ways are more exotic than going to Africa, because you're seeing things that maybe in your backyard, but you've never seen because it's so tiny. And with the technology and the lenses, we can zoom in. One of my favorite BBC ones is Life in the Undergrowth, which was just sort of backyard stuff that just blew me away. So but that's me. I might have more of a fascination with the less charismatic microfauna, but to me, it's pretty charismatic. Thanks for the question, though. That's great.
Yeah, I agree.
I'll keep working on it.
There you go. I just finished watching your Ride and Dance Water, and I heard you quote one of my most favorite hymns, "This is my Father's world." And there's a line in it that just resounds with me. It says, "In the rustling grass, I hear him pass. He speaks to me everywhere." And I think that's so much of what's captured with what you're describing here. And, a lot of times in our personal lives, we often think of what's normal and we try to define normal, and normal is just a really weird concept, because if somebody else from another part of the world comes to meet us, they see all the wonder that we get so bored with so easily, and it's not easy, Storytelling Ron, he, I think, went on and he has this next comment: "I feel evolution teaches us mundane things and ignores the complexity and unique traits of creatures that we rarely learn about their unique abilities." And I focus in at the very end with that word unique, because, there's no such thing as very unique, right? Either it is or it isn't, to realize everything has a purpose.
Right. And the only liability that these mundane things have is that they are common, and they're anything but mundane. But because we see something more than one, several times, was like, oh, been there, seen that, ho hum, it's boring. And I just think that is more of a problem with us, and we need to just get out there. The other thing about this is that even though many of the evolutionary biologists are discovering all of these amazing things, and they're often wowed by them, but there's no one to thank. Thank my lucky stars, thank it's all just time, chance, matter, and energy grinding away, and I'm like, do you not see the teleology? Do you not see the complexity? Yes, they see the complexity, but they axiomatically assume that rather than actually showing how evolution can actually build these complex, it only select for them. It, in natural selection, selects, exactly. It selects. It doesn't design teleological structures and symmetry. One of the things that I wanted to talk about, unique abilities and things like that, complexities. I tell my students this a lot. Everything we see, plants and animals, if it's not a term, is has got architecture, whether it's a fern leaf or a fern sporangium, uh, whether it's something simple, looking simple, if it's got symmetry, it's got teleology, it's got purpose, that is architecture. And when you see architecture in a building, you know that it was designed with that intent. The buildings don't look like tumors. Buildings have symmetry. Buildings have both internal and external design features. And that's the thing that I think a lot of people overlook is they think, oh, there's homology between this protein and that protein. Well, let me just use an example of like in architecture, real architecture versus biological architecture. Think of a protein as a brick. And there is information that is necessary to manufacture a brick. You've got to get the right dimensions, the right material, and you make a brick. Think of that as a protein. And you might have homology between this protein and a brick in this building and a brick in that building. Great. Same recipe for the brick. But the recipe for the brick is not the recipe for the building. And when you're talking about macroscopic metazoans, I've disappeared. When you're talking about the architecture of animals and plants, that symmetry, that architecture is a different set of information than the actual proteins. These are gene regulatory networks that orchestrate the development of these complex, how the cells divide, when they divide, how the cells differentiate. Everything is programmed, and it's like orders of magnitude more complex and different than the instructions to make the brick. You can have a same kind of brick in a Georgian architecture administration building in the University of Georgia campus and an ugly little brick rancher. Same brick, totally different architecture. And they just say, well, homology, brick. Oh, yes, but that's not information that builds the building. That's a different set. And so when you look at plants and animals, I want all my students, as well as non-students, to see the glory of God in the symmetry and the complexity, not only of the architecture, but the functionality. It's not like a sculpture of a fern. It's a fern. It's a fern that has real dynamic sporangia that flings spores like a catapult. And the mechanism of that, I just scratched the surface. It's more complex than that. There's more complexity in that pappus on the dandelion and the spore sprinklers. We're just scratching the surface. The complexity, my dad said, and I'll end with this unless there's more questions. My dad said, intelligent design, the understatement of the century. And I think he hit the nail on the head. It is the understatement of the century.
Yeah, we have a few more questions here. Would you be able to field those last few ones, you think?
Yeah, if it's not too, yeah, sure.
All right. So Jake says, thank you, excellent. What do you think would happen to water striders if they had some detergent on the water breaking the surface tension?
Water breaking the surface tension. That would probably cause a problem. That would be something that you could experiment by grabbing a few water striders and put them in the sink and doing that. But yeah, I would think that it would have a problem.
Yeah, I think that's pretty.
Especially if that detergent gets on to the hydrophobic hairs. Yeah.
Yeah, and then The final
All
question I
right.
think we've got here is, would it be correct to say that we have never observed a mutation which produced a new useful structure which was then passed on to the next generation?
Well, that's tricky, but yes, I would say that if it's truly now, how do you define mutation? If I say it's a mutation like a random literal error, a deletion, addition, and a frame shift, these are real mistakes, real typos that happen with DNA polymerase messing up. And someone says DNA gets shuffled around, I think there's a lot of genetic mechanisms implicit in their design of shuffling the deck. When you think of gene recombination, I don't know what evolutionists calls it, but there are mechanisms in which things are rearranged and little genetic domains are rearranged, and I wouldn't call those mutations. Now, in one definition, it might say, well, things got changed, it must be a mutation. But if it's by design, it's not a mutation. So, but I would say that any true chaotic mutation, where it's literally a typo, is never going to produce a useful teleological complexity. It doesn't have to be an irreducible complexity. There's things, I think we bandy that term around too much. There are lots of complexities that are reducible. I mean, I'm reducible. I'm a complexity, and I can have my arm removed, and I'm reduced, but I'm not an irreducible complexity. A car is a complexity. A lot of things on the car are designed, and you can knock the hubcaps off. You can knock all sorts of things off, and it still runs. So again, be careful with the word irreducible. I didn't say. I don't think you didn't use that. That's sort of a tangent that I'd like to address. But it's. I would say mutations. Yeah, they do not. If I would say that anytime you have mutations, don't generate novel, complex, specified information. Okay, it may add DNA. You might have a gene duplication, but that's not adding information. That's just copy and paste. That's just gene duplication. You're adding DNA, but you're not adding novel information. So, and there can be back and forth on that, but yeah, it's like giving a kid, giving, say, a drunk a pen and expecting that drunk, as he falls down on a sheet of paper, to draw up plans for the Taj Mahal. It's like, that doesn't happen. It's so obviously designed, and I think you just have to axiomatically just say it's not designed, and plug your ears, plug your, see no design, hear no design, speak no design, but they really don't have a leg to stand on. One last thing that I wanted to say, and that is, we as Christians, as we explore the natural world, don't just think of the glories of creation as an apologetic tool. We often use the complexities that we see in nature as an apologetic tool to say, see, Darwinism can't do this, mutation can't do this, and go on and keep adding arguments. But it's not just an apologetic tool. We need to celebrate creation as Christians and look at it and worship, and not just figure out, oh, what's the next new argument I can beat an evolutionist over the head with. Yes, we do need to take Darwinism down, but we also need to just glorify God as we study and unveil the glories of His creation and give Him all of the praise for it.
Yeah, I couldn't
That's
agree
it.
more with you, Dr. Wilson. I was reminded of between the comments that I'm reading through this presentation and a comment you keep reiterating echoes what the Apostle Paul wrote in the first chapter of Romans, where he listed how, it's easy for us to ignore the Creator. And it goes through the long list of all the things that they did in that culture, in that time period. And it's tucked in there at the very end of, I don't remember which verse, towards the second half of the chapter. It says, neither were they thankful. And, I think it's that thankfulness component that we as believers have the privilege to express. The unbelievers, I mean, they come from a different worldview, a different perspective. He gave it to all of us, but we get to worship. And just saying thanks it's amazing.
Mm-hmm. Amen.
So, all right. Well, I had a great time tonight listening to Gordon, and I hope I get a chance to
all right.
follow up with the rest of the ride and the dance as they come. Thank you for this fascinating discussion. As a reminder, we want to encourage anyone watching to check out the Logos Research Associates website. On the website, you can learn about the various research efforts of all our research associates and ambassadors and find information about their publications. Please take a few moments, browse the website, and consider supporting one or more of the exciting creation research projects that are underway through Logos Research Associates. While you're on the website, please be sure you have subscribed to the Logos email list so you can receive notifications about our other upcoming presentations. When you subscribe, you will also receive the Creation Evolution Headlines newsletter. We want to let you know that a recording of this video will be made available on our website, so you can share it with others who may not have been able to join us live tonight. Our next presentation will be on Wednesday, July 9th at 9 p.m. with Dr. Mark Horsemeyer, professor of engineering, giving the talk Multi-Scale Design Paradigm, Studying God's Creation from a Creationeering Perspective. You're going to enjoy it. Every time I get to be around Mark, it's always an encouragement and he sets the bar high, with where we see things moving, and very much like what Gordon was talking about as well. So we'll hope to see you on Wednesday, July 9th at 9 p.m. Eastern Time. I'm going to close us now in a short prayer. If you would, join me. Let's pray. Heavenly Father, in one word, we say thanks. Thank you for this creation that you have given to us, that we can plumb the depths of it and forever just stand in awe and in an appropriate response. The only response is that of thanks and worship to you, because you alone are worthy, because it was your great mind on display for us. And what a privilege it is. Thank you for this time tonight. Thank you for how you have put together my friend Gordon with his passions, his desires, and his ability to communicate. I ask that you would continue to bless his efforts in the series and his research and all that he's doing with his family and friends and all the work that he does. Again, thank you for this time. In Jesus' name, amen. Thank you all for joining us, and see you in the next broadcast.